Ahora sí, ¿verdad? Faltamos algunos, ¿verdad? Todavía veo. Bueno, esperamos unos 2 minutos y comenzamos. ¿Les parece bien? Ok. Bueno, vamos a comenzar con nuestra charla de hoy. Es un gusto para mí presentarles a Rebeca Hernández. Ella es investigadora de aquí de Sinvesta Irapuato, recientemente integrada a partir de marzo, ¿verdad? De este año. Y pues les voy a contar un poco de Rebeca. Ella es bióloga por la UNAM. Hizo su maestría también, perdón, por la Universidad de Aguascalientes y la maestría y el doctorado en la UNAM, en el Instituto de Biología. Y ha trabajado como investigadora postdoctoral en la Universidad de California en Riverside. Y bueno, su trabajo se enfoca en la evolución de plantas a escala micro y macroevolutivas. Y bueno, pues muchísimas gracias, Rebeca, por estar hoy con nosotros. Y esta charla les cuento que va a ser en inglés. Ya las siguientes charlas desde hoy van a ser en inglés. eh, Dado que aquí tenemos de invitado a Peter Stadler, que mañana va a estar dándonos uno de los cursos. Entonces, bueno, es una oportunidad para practicar nuestro inglés. Gracias. Adelante, Rebeca.
Buenos días. I'm switching to English now. Thank you for coming today. I'm gonna talk about most of my PhD dissertation and some follow-up projects that I have. And I will talk about how I have been combining genes, fossils, and some probabilistic models to understand plant evolution. So to start, I want to present to you some of the biodiversity patterns that we see globally. For example, we have here some of the 3 maps that show biodiversity in birds, mammals, and amphibians. And this is represented by species richness. How many species of birds, mammals, and amphibians are in the world? And so as you can see here in these maps, the warmer colors, that means red in this area of the gradient, this means more species, and the colder colors mean lower diversity. Okay, so less number, a lower number of species. And so what we can see here in these 3 graphs is that in the tropics there are more species, and we can see as well, I didn't put the title here, but this is vascular plants, okay? And so here in the graph, what we are seeing here is that lighter colors mean more species. And as you can see here, for example, in the northern part of Africa, in the Sahara, there are less species in general, right? And so here we have soil fungi, and also we can see the same pattern. And so this pattern means that there are different lineages, different groups of organisms that share a pattern that is higher diversity or higher number of species in the tropics. Okay, so this is a very general pattern, and we call this pattern the biodiversity gradient. Now, we can also see a biodiversity pattern with elevation, right? And so here we have in the x-axis the elevation in meters above the sea level, and here we have the species richness. And you can see here in these silhouettes the type of organism that they are measuring. Now, what we can see here is that some lineages have very low diversity, right? Very near the coasts, but higher diversity in some kind in the middle of an elevation gradient. And so this happens with ferns, for example. So there are more species of ferns at middle elevations. But in general, what I want to show you here is that there's also patterns in elevation. There are patterns in latitude. So the global, these global patterns that I show you, okay? So these are general biodiversity patterns. But this is in the present. So now we are measuring or counting species in the present, but what happened in the past? We also find biodiversity patterns in the past, okay? And so here what we can see is the number of morphospecies. We cannot say that there are species in the past or with pollen samples. And so these authors here measured how many morphospecies based on pollen, fossil pollen, were found in Paleocene, Eocene, and Oligocene, and so on. And so what we can see here is that the number of species increased. Some were in the middle of the Eocene, okay? And so that diversity declined towards the present. So this in the past is also a biodiversity pattern. Now, what I want to show you now is that this pattern of biodiversity was kind of correlated or just related to an abiotic factor. And so here they are measuring oxygen ions. And so this is somehow related to temperature, global temperature, okay? And so this is some proxy to measure past temperature. And here you have the Eocene thermal maximum. And in the Eocene, they found a higher number of morphospecies. And so they sort of relate the number of species with the temperature increase. And so this is in the past. So we have biodiversity patterns in the present, biodiversity patterns in the past. And so we can analyze past and present patterns to understand how life evolves. So we can measure and we can count and we can make correlations, but also we can try to figure out what are the process that underlying those patterns, okay? The evolutionary processes. And so I'm interested in these three factors that are influencing biodiversity patterns. Dispersion, for example, if you are in the coastal area of the continent, and then you acquire some mutation and you can tolerate higher altitudes or higher elevation, then you are dispersing upwards in the gradient, in the elevation gradient, right? And so there are other factors, for example, biotic and abiotic. And today I will talk about my projects related to the biotic factors and dispersal. Right, and abiotic factors can be, for example, the tolerance in temperature that we see in the morphospace in the morphospecies of pollen. Okay, so why are there so many species, and especially why are there so many species in the tropics? And that's my main question. The question that I'm most interested in answering, and I try to answer that question. Measuring time, the dispersal globally, and also measuring the phenotypic evolution of some plants. So another thing that I want to stress now is that biodiversity is really, really complex. And so to understand biodiversity patterns, the processes underlying that, we can do that through different approaches. And so my approach is to using phylogenetics and making independent analysis. So my study system and the group that I like, that I really like in plants is this order Malvalis. And it's really interesting because it's pantropical, so it's distributed across the tropics in the world. It has a high number of species, more than 6,000. And there are many ecological interactions that we know. For example, pollination, association with other plants. For example, some of them are parasitic plants, etc. And another important thing about this order is that the fossil record is really abundant. And so there are many fossils. So my question first to understand the tropics is to use this order as a representative of the tropics in the world, okay? So this is a representation analysis, and I am drawing hypotheses about the tropics based on this order, okay? And so it's composed of 10 families, and these are the flowers. These flowers are beautiful, but also every family has its own interesting thing, and most of them are distributed in the tropics. So my first question was when and how the families of Malvalis originated. And so I searched in the fossil record and I found more than 100 fossils from all kinds of fossils, for example, pollen, some flowers, and here you have another flower, and wood, and so many leaves. And so in the fossil record, there are so many different ways of describing fossils. So I made some filters and I get 80 fossils that I could actually use. And so, for example, what we have here is this stratigraphic chart. And we can see that there are fossils here, for example, North America, and also some fossil here in East Asia, well, in Asia, in China maybe. And so there are two occurrences of one species or one morphospecies. And so for this project, I included both. So I included anything that was in the fossil record in different areas, okay? Different areas, same species or same morphospecies. And so I used this model called fossilized birth-death process to integrate phylogeny of extant species and the fossils into that phylogeny, okay? And so for that, I'm gonna present some of the parameters of this model. For example, we have the speciation rate and extinction rate, and this is important. These parameters are important because a phylogenetic tree, if you remember, is a history of bifurcation events, and every bifurcation event is a representation of a speciation event, right? And so we can measure that, and also we can have, for example, the probability of sampling an extant species, but also the fossil recovery rate. Some fossils have less probability of being recovered, and so that's also an important aspect of this model. And so here we have an example of a phylogenetic tree, and all these red circles represent fossils. And so here is like a representation of what happened in reality. And so we can sample fossils from the fossil record, and we can see that some of them are ancestors to living lineages, but some of them are ancestors to extinct lineages. And so this is another important aspect to consider. Not all the fossils that we found in the fossil record are actually the ancestors of living species, okay? And here is a graphical model. Oops. This is a graphical model of how these parameters here are estimated. And so we have, this is the speciation rate, extinction rate that I mentioned before, and this is coming from a prior distribution or a prior distribution, a probability here that we can set. And from that prior distribution, the algorithm will draw some of the estimates and test hypotheses about this particular parameter in every generation that we run in our Markov chain. Okay, and so this is the model. We need to estimate these parameters right here to obtain a phylogenetic tree with fossils in it, okay? And so this is quite complicated. And so because of that, we rely on this MC3 algorithm, Metropolis Coupled Markov Chain Monte Carlo. Maybe you have heard about this because they're very popular, especially in Bayesian analysis. And so this algorithm, what it does is that it takes the state or the value of one parameter, and then it changes, and it changes constantly. And so at the end, this is drawn from the prior distribution. And at the end, having many iterations of this sampling from the prior, we have this posterior distribution right here. But this is only one chain with one parameter. And so it does this with all these parameters right here, the circle parameters, for many generations with multiple chains, okay? And so Metropolis coupled are many chains, many Markov chains that have different components, right? And so the most important thing about this is that we need to obtain parameters that are representing our reality the best as they can, and this can take a long time. And so this is implemented, sorry about this, implemented in this program here that's also very popular for Bayesian evolutionary analysis. And so the result here is this phylogenetic tree that we have formal valleys. And so as you can see, these red lines are actually branches that represent fossils. And so, for example, we have this bone here that actually is over here somehow. And yeah, and so the fossils that I presented earlier are sampled here, okay? And so the molecular database consisted of 8 genes and 230 species. This is not a lot, but this is what it exists in molecular databases such as NCBI. 8 fossils are right here. And well, one of the results is that all the families originated in the Upper Cretaceous. And so that represents 20 million years of origination of these families. And that's between 90 and 70 million years ago. So another thing that's really interesting using the fossil record is that they not only say the age of when these plants existed, but they also say where. And so that's information that's really, really valuable. And so I used this information to ask what's the geographical history of the order. And so to reconstruct the space occupied in the past is quite complicated. And so we have some models to understand this in the past, right? In a probabilistic realm. And so, for example, here we have a model of speciation within an area. And so let's say that there's a lineage living in this area here. There's an speciation event for something happened, and this lineage diverged in two. But these two lineages are occupying the same space, right? And so there's no change in space. The only thing that changed is that there are two lineages that come from one ancestor. And so the next example is where there's speciation, but what happened is that there is the range of the daughter lineage changed here, and the rest of the lineage stayed in this area, right? And so these shaded areas are the present distribution of the lineage of the daughter lineages, okay? And so here what we call this is not extinction, but it's something called extirpation. And so in biogeography, you will see that there are extinction, which is completely death of a species or a lineage, but there's also something called extirpation, which means that one lineage or one species is no longer living in an area, but it's living in another area, okay? And so we are modeling extinction, extirpation, and speciation as well. And so there's another possibility that doesn't involve diversification or doesn't involve divergence of the lineage, and this is a anagenetic change. And so anagenetic change means that it's a lineage that changes the region, but doesn't split in two lineages, okay? And so that's what we call anagenetic change. And so, for example, this is represented by the range expansion. And so here we have an anagenetic event. The lineage didn't split, just changed the range, okay? It expanded. And we have also a range contraction. And so that doesn't represent a cladogenetic event. That means a bifurcation. And so bifurcation is a speciation event or cladogenetics, okay? Cladogenesis. Now, so we have these models here, and what we actually do is estimate the past ranges throughout the phylogeny. But I want to point out that we are seeing here 0 and 1. This is the area, right? One area is 0 and another area is 1. But as you can see here, there are more than 2 states. One state is 0, 1, 1, another state is 0, 0, 1. And so these are the actual states. And so it's more complicated than just one state or binary state, right? And so we have 2 areas, but we have a state that is absence, absence, and present in the third, in another area, right? And so we have areas, and we have a state where these species or the lineages are living. And so for this, to model these past distributions or ranges, we use something called stochastic character mapping. And this is because sometimes we have the states at the present, and we can put that in a phylogeny, and then we can estimate the past, like in an ancestral state reconstruction. But in the meantime, in these branches, something is going on. And so to make it more real, to model life a little bit better, we can also see changes in the branches. And so if you have a phylogeny, you can estimate the changes in the branches of a phylogeny if these branches are representing time, okay? And so in time, we can also estimate the changes until the present, for example. And so stochastic character mapping is an algorithm that estimates first the nodes in a phylogeny, the past, the state or the ancestral range in the ancestral immediate to the two present species, and so forth until we reach the root of the tree. And after that, there's another step to use the likelihood of this ancestral state to estimate this node right here, and so forth. Okay, and so there are two steps. First, from tips to root, and then to root from the tips. And after that, we are validating this, the changes in the tips and the root, but estimating this intermediate or this history of a lineage that didn't split. Okay, and so this is a basic component that characterizes stochastic character mapping. Stochastic character mapping is used for traits as well as for biogeography, but for biogeography is quite convenient. And so at the end, we can see the changes in areas along a branch. And so for every bifurcation in a tree, we can calculate the likelihood, as I said before, and we can do that for every single one of the models of distribution. And so these right here are the models of distribution. So at the end, we have a matrix with the probabilities of a model of distribution and the probabilities of the ancestral area. Okay, and so what I did was including a little bit more. I increased the number of species in my phylogenetic tree of Malvalis, the same fossils right here, but the thing is that I used the geographical distribution of those fossils. And so this phylogeny includes now time and living species and fossils, and also the area that these lineages were living. And so in this case, I used non-geographic areas that sort of represent continental masses, but not completely. As you can see, there are not more. And so because of the past, and because Malvalis is quite old, and the world was not similar as the organization of the continental masses was not as it is today, I also included 4 slices of time. And so the only thing that differs from the stochastic character mapping that I showed you is that there are mappings that have more probability than others. And so if a region like this is quite close, then the probability of dispersal is higher. That's it. And so here, this is the result. And as you can see, this is the same phylogenetic tree that I showed you with some branches that don't reach the present, which are the fossils. And so here you can see these areas, and these areas mapped here in the global map. and some combinations of possible ranges of this species. And so what we can see here is is interesting because, for example, here we have the family Malvaceae, and one of the probability of this of the ancestor of the family is higher. It is higher for the distribution of North America. And this is quite interesting because most of the species of Malvaceae don't live in North America anymore. And I'm not gonna get into details, but the thing is that I did a test without fossil record, and the story biogeographical history is not similar. And so my conclusion here, just very quickly, is that fossils influence a lot in the results because there's evidence of past distributions. And that makes sense because the tropics were much higher in the past because of the temperature, right? And so the distribution of these fossils right here is in a zone called boreotropical, right? And so it's found so many fossils in North America that show this, and not only for Malvaceae. Okay, so this was really, really cool and exciting. And so now we have a time estimate of the origin of the families of Malvaceae. And so this is a representation, like I said, from the tropics. And so this is an evidence or a hypothesis of when the modern tropical biomes originated, right? And so if we count this if we compare this result with other species of tropical plants, we can see a similar pattern, and that's been already done. So now, so now we have a hypothesis of how these plants distributed globally, and now we can make some more fine hypothesis of each of the lineages in Malvaceae. But now what I want to show you is how phenotype evolves, okay? And I'm gonna talk about Malvaceae in particular, and maybe a little bit about another order that's really interesting as well. So another biodiversity pattern is that Malvaceae is a family with higher species richness in Malvaceae. And so the number of species per clade is another biodiversity pattern. And so you can see in in the tree of life that there are some lineages that have more species than others. And so it's not just about the space, but it's also about how many speciation events occurred or how much extinction occurred, okay? And so it's about the process of speciation and extinction and the dynamics between them that we can see these other patterns of biodiversity. And so Malvaceae within Malvaceae is quite interesting as well because of phenotype. So you can see some trees like this that are adapted to seasonally dry tropical forest like here, and some flowers that are really weird like these ones, and these are from Mexico. This is from Ecuador. Anyway, so there's so many interesting things about the phenotype of this family. So I went to field, I collected some plants, I extracted DNA, and this is me with one of those trees and so many exemplars of these plants. And at the end, I got this species tree for the family, and I was very happy with this. But so the underlying processes that generate this tree are quite complicated, right? And so here we have the gene trees. And as you can see here in these shaded lines, is that there are some genes that are that don't have like that don't share the same history as others, right? And so we can see here that some genes are telling us that some lineages right here are also closer to some lineages right here, and that doesn't that's quite complicated to understand, okay? And so what I wondered is what's happening, what's going on? And so to estimate the species tree that I showed you, I used coalescence, multi-species coalescence models here in this program called Astral. And so this is taking quartets that are formed by 4 terminals. It doesn't matter if these are species or just branches, but it has to be formed by 4 terminals. And here you have X1, X2, Y1, and Y2, right? And so we have these 4 terminals, and these 4 branches have to form a tripartition, okay? And so here you have a tripartition. We have these 3 branches that are linked by a node called U. And so this is a tripartition that is fully resolved, and it's a rooted tree. And so we are very happy, and we want to find more of this in a phylogeny to build the species tree, okay? And so here, for example, this is not a tripartition, this is a multipartition, and this is called a polytomy. We cannot resolve this with the genes, okay? And so we are not taking this into consideration, and we're scoring the number of quartets that we found forming a tripartition, okay? And so this is the calculation is for all the branches in the phylogenetic tree of every gene. And so the algorithm is counting all the tripartitions from quartets, okay? And so at the end, we want to maximize the number of quartets in our database. And so with that, we can get something like this. And this is the species tree. And so I'm not gonna show you, and I'm not gonna get into the details of the support, but all these numbers or these tiny dots that you're seeing above the branches are the scores of the quartets. And some lineages are supported by many quartets, but some lineages are not. And so at the end, we still don't know what happened. I'm just gonna talk about this in a second. So what we found, because of this sort of discordance in the phylogenetic trees, we found that this is due to a high proportion of introgression in the past. And so introgression is the genetic flow between different species. It's like a type of hybridization event, but it doesn't form hybrids. It's just the genetic flow between one lineage into another, okay? And so we have found a high proportion of introgression, and these branches right here, these black arrows, mean that there's introgression between distantly related lineages. And because of this, we can say that this is ancient introgression. And so some of not in the present, but in the past, the ancestors of these lineages were sharing, there was genetic flow between those lineages, okay? And so this is the reason why we can see so much phylogenetic discordance in different genes. Also, I want to mention that those genes are from the nucleus, and so it's not plasmid inheritance. And another thing is that this is just a model testing to see how much introgression versus incomplete lineage sorting is there, causing phylogenetic discordance. And so it's more about introgression and not so much about incomplete lineage sorting. Okay, so the interesting thing about introgression is that it has really important introgression, important implications in evolution. And I'm gonna show you an example that really makes me happy, because it's about this. And I don't know if you have seen this figure here. Is it familiar to you, some of you? No. Sí, yeah, the Little Prince, El Principito. So some of you have seen, did you say that? I didn't hear, sorry. So anyway, these are the baobabs. And so in the Little Prince, these trees were really bad and they were scared. And so, but anyway, the point is that baobabs are really cool plants, and I really am very excited about to present this paper. And I honestly take every opportunity I have to present this paper because it blows my mind. Anyway, so instead, baobabs are not just interesting because they are high, weird looking trees, but also because they have really beautiful flowers that are dependent on pollinators and very specific pollinators, bats, for example, and moths. Okay, and these are distributed not in asteroids, but in Africa, Madagascar, and one species in Australia. And so the thing about this is that there was an introgression event that changed the flower, and because of that, it changed the pollination type. And this is what I really like about this paper. And if you are interested in this, I really encourage you to do. If you're not interested, you will be interesting because it's an amazing paper. And so the point is that here you have Adansonia digitata, which is the baobab, the common baobab. And so there was a introgression event here that changed the pollination type. And so as you can see here, these species are hawk moth pollinated, and the flower looks something like this, but something happened in the past. And so the genes that are expressing the floral phenotype and in, for example, color and the arrangement or the architecture of the stamens changed. And so this lineage here turned into a mammal pollinator type. And mammal is bad pollination. And so the color of the flower changed, and because of this, the type of pollinator changed. And the implication for this is really interesting because it changed a biotic interaction, okay? And so, yeah, this is really interesting, an interesting study. And this is was an inspiration for me to do a follow-up of my project about Malvaceae. Oh, and another thing is that this is a case of adaptive introgression, okay? And adaptive introgression means that their adaptive introgressed genes gave the ability to adapt to something to the receptor lineage, okay? And so this is one evolutionary process that that's really, really cool. And so this is one example of that. So my question now was if the introgression could explain some of the floral phenotypes of Malvaceae. And so as you can see here in these pictures, there are four different flowers of Malvaceae. And so here you have the chocolate plant. And so this is a flower. And this is another species, and so on. And so one thing that is interesting is that some species of Malvaceae have a specific arrangement of these stamens, okay? And so we have here these two types of, well, this type is the most common type of stamen. The organs that generate or that produce the pollen, okay? Pollen generating organs are the stamens, and the stamens are divided in two parts. This is I forgot the name in English. That's not important. The important thing is this, because this is the anther. In the anther, the pollen is generated. And the anther in most angiosperms is divided in two thecae, okay? In Spanish is tecas, two thecae. And so the point is that in some Malvaceae species, we have this two separate tissue stamens with one theca each, okay? And we call this monothecate anthers. And so the point is that some lineages that are distantly related have monothecate anthers. And the hypothesis is that this division marked an important or it's related to bad pollination, okay? Related to bad pollination because many species in Malvaceae are bad pollinated. And those that are bad pollinator, most of the time have this arrangement of the anthers. Okay, and so I wanted to see if the introgression is related to this evolution of the anthers. And so I used a phylogenetic network for this because, like I showed you before, there's a lot of introgression, ancient introgression in the family. And so I estimated this network with this program right here that uses maximum likelihood to estimate the networks. And what it does is that it takes a phylogenetic phylogenetic network like you're seeing here. And so these are the species, our taxon sampling, for example. And so as you can see, there are two introgression, or let's just call them reticulation events. And so we have two reticulation events right here and another one right here, right? And so this is a phylogenetic network that's simulated there. And what it does is that it prunes one of the parents of each reticulation event like this. And so this, now you can see that there's a shaded branch leading to this and a shaded branch leading to this node right here. And so this is a displayed tree. What that means is just that it's building phylogenetic trees, okay, out of a network. And so we are estimating the number of reticulation events in our sampling with a multiple sequence alignment, okay? So we have a multiple sequence alignment, we have many genes, and we can find these networks here, and there's an algorithm that prunes the one parent for each reticulation event, and then we can estimate the likelihood of that phylogenetic tree. And so, yeah, one of the parental branches is pruned. We can calculate the likelihood of this displayed tree, and we can do that for every set of species and set of genes. And so for Malvaceae, I found 8 reticulation events, and this is a lot. And so this is just a subsample of my sampling for Malvaceae. And so I only included these 16 species, and I found these reticulations. This is really overwhelming, honestly, because it's a lot to process, and it's quite complicated to understand. But what I want to show you is that I'm interested in the evolution of this group right here, because this is a group that has these monothecate anthers, as well as this species or this lineage that's represented by the durio or the durian. I don't know if you know about the durian, but it's a fruit that's very smelly in Asia. Anyway, so I combined this phylogenetic network. What time is it? Okay. Combine this phylogenetic network with an ancestral state reconstruction. And I use these programs right here. And the point is that the states in the tips can be used to calculate the likelihood of the ancestral state at the nodes. But this also includes another component where there is a proportion or a proportion of the contribution of this lineage to the state or the ancestral state of this lineage here that's the daughter of reticulation event. Okay, so it's an ancestral state reconstruction that's commonly used, but it includes the proportion of each parent for the reticulation. And so in a phenotypic space right here, we have the time, we have here the present, and we have the states or the phenotypic state of each of our living species. And here we have the state that's a combination of something in the middle of two lineages right here, okay? And so this is how it looks like. And so for purposes of this talk, I am only gonna focus on these three reticulation events, are 6, 7, and 2. And so these are the most important nodes that I want to show you today. And these are the how I am gonna find if the introgression is involved in the evolution of the flower. And so what I found is that I tested many characters, but I'm gonna show you just this one, which is the number of thecae. And so as I showed you before, this is the monothecate state, and these are possible ways of being dithecate, 2 thecae, okay? And this is normal for angiosperms. And so what I found in this node right here, that is the ancestral node, or represents the ancestral lineage of my focus group right here, is that the ancestral was monothecate, and this comes from the reticulation with the lineage of the durian. And so this answers a little bit. It might be possible that in fact, yes, the introgression right here in the ancestral lineages of the durian and this group of species here, that includes the baobab, of course, was the genes that express monothecate anthers was introgressed into this lineage. Okay, so that's kind of the conclusion, but I'm still gonna make some more tests. So in conclusion here, the reticulation can explain the acquisition of some of the monothecate anthers. So to conclude this part of the talk, the Malvaceae family is characterized by many events of introgression, ancient introgression. It can be many introgression events that are adaptive. And so we have one with the baobabs, we have another one with the monothecate anthers, and I don't know, maybe there are many, many more. Okay, and so monothecate anthers may have been transmitted to the lineages through introgression, okay, to the lineages that are now characterized by monothecate anthers and that are pollinated by bats. So to come back to my one of my first slides, I showed you today that how I model dispersal globally for the order Malvaceae and some of the phenotypic evolution of Malvaceae. And so this represents dispersal and biotic factors, okay? And so the complexity of biodiversity is really, really high, and it's represented by complex models, okay? So for example, the fossilized bird death process is a model that's quite complex, but we cannot really optimize these models unless we have really powerful algorithms, okay? So that's my algorithm conclusion. And well, maybe for next time, I will show you this model here that I designed for colleagues, and that is related to abiotic processes or abiotic factors that influence biodiversity in another group of tropical plants. But thank you so much. I'd be happy to answer your questions.
Thank you very much, Rebe. It was really nice talk. Questions you have?
Well, congratulations for your wonderful presentation. My first question is which is the best methodology to extract the fossil DNA?
Okay. Good question. I didn't do that because it's not possible. For most of the really ancient fossils, it's really not possible. For some archaeological samples that are not millions of years old, might be possible. But I included those with a model, right? So it's just that I said this fossil belongs to this group. Do whatever you want to do, and model the branch length based on the branch length of the living species. And so I had the molecular or the DNA for the living species, and I modeled a phylogenetic tree. And after that, I said this model belongs to this group, and the branch lengths were modeled for that. And so it's like just including fossils, but not the DNA, right? Mm-hmm.
And my second question is is there some database with a fungi fossil register?
I don't know. There's a big database called Paleo database, and that is quite comprehensive. It includes so many organisms, but I don't know about fungi. And so it might be there, but there are very, very few. And so I am working with a colleague of us, Christian, who works with the genus Amanita. And it's really complicated to date the phylogeny because there are no many fossils. Not just many, there are two for the whole agaricales. So it's, yeah, no.
Thank you so much.
Hola, buenas. Muchas gracias por la presentación. Te quería hablar bueno, tengo una pregunta sobre tenemos también plantas cultivables, como es el caso del cacao. Entonces también el proceso de la domesticación podría estar influyendo en la diversificación de las especies.
Sí, claro. La cosa es que en estos análisis que son a gran escala, usamos genes que estén compartidos por muchas especies que divergieron hace millones de años, como presenté. Esos genes, como están conservados, son tan fundamentales que la domesticación no les ha afectado tanto. Entonces, de manera comparativa, no podemos ver en estos análisis el efecto de la domesticación. Pero si tuviéramos una filogenia de todo el cacao y de las especies o de diferentes accesiones o diferentes cultivares del cacao, lo veríamos. Eso sí.
Gracias.
Pero no hay que hacerla.
Sí.
Hi, thanks for the talk. I wanted to ask about the slide on which you show how to get the model parameters. I think there was a probability estimation
At
the beginning. Yeah.
Oh, yeah. This is gonna take a while.
It's okay. Yeah, over there.
This one.
The next.
Mhm.
Yeah. Could you help me understand what the
Yes.
samples mean in that case on the prior distribution?
Right. So this prior distribution means that we have values from this range, from this limit to this. And so, for example, this could be 1 and this could be 0. Okay. And so the samples in the Markov chain is, for example, taking this value right here. And so because this is 1, maybe this is 0.8 for this parameter. And so at the next step in the chain, you will take, for example, 0.7, okay? And so it samples from this distribution of possible values, okay? And so every step in the Markov chain, you take a value, if this value has a higher higher likelihood, it stays there. If it doesn't, it changes, okay? And so so every step in the Markov chain is taking numbers or possible values out from this prior distribution, right? And so this is a probability distribution that it's the first step that you will have. And so, yeah, at the end, you will get something like this with very low probability for these values right here and for this, but at the middle, you will have a high density or more, the probability will be higher in these values right here in the middle. Well, not always in the middle, but yeah.
Thank you so much.
Ay, hola.
Hola.
Bueno, tenía una duda sobre los criterios que se utilizan para filtrar los fósiles. ¿Cómo se hace esa filtración?
Sí, interesante, buena pregunta. La filtración incluye, I'm gonna answer in English if that's okay. I forgot. But the fossils can be filtered based on if the fossil is comes from a dated stratified area or a formation, and so you have a formation with a stratigraphic date that this date has to be very well done with carbon and ions and all that kind of stuff, or some indicators like foraminifera. Okay, and so this formation has to be very well dated, and it has to be included in the fossil, in the stratigraphic chart, for example, and it has to match the stratigraphic chart, okay? And so that's the first step. And so if someone finds a fossil that doesn't have a formation, we cannot include that because we don't know if that's true or not. Another important factor for the fossils is that we need to have measurements, right? Comparisons. It's like a morphological comparison of the fossil and which group this fossil belongs to. So for example, if we have pollen fossil, we need to see the structure, the actual fossilized structure to know this pollen is really a pollen, first of all. And second is this pollen belongs to this group. It doesn't matter if it doesn't match a species or even a genus, but it may be a family. And so that's another thing. And third, it has to be, this fossil needs to be placed somewhere in the world. So we need to be sure that this fossil, we can get there and we can see the fossil. So if someone has a fossil in your houses, that doesn't count. Even though we have the stratigraphic, the formation, or we know what fossil is, but if it's not in a collection, that also makes us suspicious. Mm-hmm.
We have a question online, or a couple of questions online before we continue with the questions here at the auditorium. So, Mari Carmen, do you want to open your microphone? There is two Maria del... Okay, Mari Carmen first and then María del Carmen.
Sí, Mari Carmen. Sí, me escucho y me veo.
Sí.
Ok, buenos días. La verdad estuvo muy interesante la plática y mi pregunta es, ¿por qué o cuál fue el motivo que los motivó, ¿verdad? A estudiar esta planta, las malvas, y a lo mejor no estudiar otras como, no sé, plantas endémicas mexicanas o plantas que tienen también más, digamos, más como ancestros antiguos. ¿Por qué específicamente estudiar estas plantas y no otras especies?
Gracias por tu pregunta y es muy buena pregunta y la verdad podría hablar todo el día de las malvaceae porque son, como dije, es un grupo fascinante. Tienen de todo, tienen mucha variación genotípica, variación fenotípica, interacciones, están en todo el mundo, pero no solamente están en todo el mundo, también hay unos grupos que solamente ocurren en Mesoamérica y mi siguiente investigaciones van a ser de esos grupos mesoamericanos y es un grupo que lo tiene todo, la verdad. Y bueno, además de que lo tiene todo, tiene muchos recursos genéticos disponibles, entonces hay varios genomas, en la familia. Por ejemplo, en la familia está el algodón. Entonces está el genoma del algodón, el genoma del cacao, el genoma del durio, el genoma de la nuez de cola en África. Es decir, hay mucha información que se puede utilizar y no me sorprende porque es un grupo fascinante. Sorry about that. I answered that in Spanish, so I'm gonna switch to English. But in conclusion, it's a fascinating group. It has everything: genes, genomes, phenotypic diversity, and it's distributed globally. It has so many weird interactions as well. And so bad pollination, which is also fascinating, and cotton, chocolate, the durian, cola nut. So there's so many economically important plants in this family. So that's the question, was why I chose this family.
Okay, thank you. There was another question, María del Carmen. Do you want to open your microphone?
Digo, ¿me
Okay.
pueden escuchar?
Sí, te escuchamos.
Tenía la pregunta de ¿qué libros o papers, ya sea en inglés o en español, recomienda para tal vez conocer un poco más sobre este tema para estudiantes que tal vez no están muy adentrados en este tema, pero pues que, por ejemplo, a mí pues me resultó muy interesante toda la presentación?
Thank you so much, Maria. The question was if there are literature that I can recommend to get introduced to this topic. And I would say that I will think about it and I will make sure that you have the links to that. There are so many possibilities, but maybe, I don't know, there's so many possible references that I can choose from. And please leave the email, or I can show, I don't know, maybe share some links to basic introductory literature, because this topic is fascinating. Thank you so much.
Okay, so I don't see more questions online. So there was one more question here.
Hola,
Hola,
discúlpame si no utilizo bien los términos.
está bien.
Hablabas de la adaptación
Ujú.
de la planta. ¿Habrá algún algoritmo o ya sea hay algunos programas que puedan determinar si hay coevolución entre las especies? Porque supongo que esto tuvo que ver con algún evento de coevolución, ¿no? Tal vez entre, por ejemplo, las que mencionabas que eran polinizadas por murciélagos y las otras que eran, creo, por una polilla.
Yeah, thanks. So the question was if this is due to coevolution and if there are algorithms that estimate coevolution, right? So yes, there are models, for example, ancestral state reconstruction type of models and algorithms there are some of them that include a correlation, like a coevolution of traits. And so you can estimate how different traits are correlated between one another and how they are evolving. And so that's already done. Another way to answer to your question is that this is a correlation. And so testing actually for coevolution is really complicated, really. And so at this scale, at macroevolutionary scales, we can only rely on correlations and these types of algorithms and models of shared or correlated evolution. However, testing for actually events and cases of actual coevolution are really complicated in plants, but there's one that's really interesting, and this is with the yucca plants and that are pollinated by a moth. And that is an example. Oh, and there's another one, figs, los higos. Figs are co-evolved with the wasps that are pollinating those fruits. And there are different scales. At different scales, people have studied that. So yeah, there's evidence for that. You should look into that.
Thanks a lot for the talk, and thanks for giving it in English, more or less for me.
Yes.
What I'm interested in is how how divergent can the various species be so that you still see introgression between them? Is there a limit if you go back in time? If they are too divergent, you don't see it?
Yes, I think so. I'm not sure what's the limit, but I'm sure there is one. For example, I don't think that we are going to see any introgression between if we have the tree of life in the first divergences. I don't think because the introgression is it's really hard to see.
You need a direct exchange of genetic material, right?
Yes, and another thing is that finding the right genes to make such comparisons are not going to show this because they are very conserved, and I think that's why. So if you have a phylogeny of animals and plants and fungi, for example, the genes that we need to use that we need to use for a phylogenetic tree for those are quite conserved, and I don't think those genes are gonna be
because the data that I have in mind is so that I know about introgression is basically all animals. So if you look at the human genome, you get Neanderthal and something a little bit older, but
yes,
that's about the limit. And same thing for mouse lineages.
Yeah, and another thing is that some organisms tend to introgress more. In plants,
it's a lot more.
Yes, and that's another thing.
Thanks.
Hola, gracias.
Hola.
Oye, ¿hay algo equivalente a los haplogrupos ahorita en todo tu estudio que has hecho?
¿Qué quieres decir con equivalente?
O sea, si ves que los haplogrupos, bueno, se utilizan mucho, ¿no? Para la parte de estudio de la genética, ¿no? En la parte humana, ¿no? Y entonces en la parte de plantas hay algo equivalente también para estar buscando este tipo de estudios, pues. Ajá, exactamente. Ujú.
Sí.
¿Sí? No lo has utilizado para tus...
No. So the thing is that for population analysis, so this is because we only have one species per lineage. It's really hard to really have the gene variation that can give us haplotypes. Okay, and so we don't have the sort of resolution in our data set that actually give us a haplotype. One thing that could be related to or marginally related to haplotypes is the coalescent based model, because coalescence is simulating the tips in the tree as populations of one lineage. But that's only for model purposes, and we don't really have the actual variation of haplotypes or making groups of populations.
Otra pregunta.
We thank Rebecca again. Thank you very much, Rebecca. It was really beautiful
Thank you.
presentation.
Gracias.
Okay, vamos a la otra vez.